Hi class, welcome to the second lecture for lecture number 9. This lecture is going to focus on the second of, second of 4, I think, different types of recombination that can occur in bacteria. So if you recall, we already went over, let me see if I can get on there. We already went over bacterial transformation, and bacterial transformation is when we have a small piece of DNA that can be imported into the cell, into a competent bacterial cell, and that little piece of DNA can incorporate into the bacteria's own chromosome. So most of the time, first of all, this is a very rare event. It doesn't happen often. Only certain kinds of bacteria can do this. And a lot of times the bacteria the genetic material, the DNA that is taken in and incorporated into the chromosome doesn't even code for anything important, right? We learned that, like in this particular example, it coded, this little piece of DNA coded for streptomycin resistance, which is an incredibly valuable piece of DNA for this bacteria, because now this bacteria has a new weapon in its arsenal, because it can now exist in a media that contains an antibiotic called streptomycin and survive, which is a huge survival advantage for this particular bacteria that has it. But understand that most of the time, transformation, again, in nature, in the real world, transformation is an incredibly rare event. And even when it does happen, the chances of it having an actual important piece of DNA that's included in that little piece of recombinant DNA is extremely rare. But remember that 1 in a billion chance, or that 1 in a 10 billion chance, is going to happen in a pool of bacteria, simply because there are so many bacteria in that pool of bacteria. Remember when we had that little microcentrifuge tube that we did the standard plate count on, had 10, I think it had 10 to the 9, which is what, 10 to the 6, 7 is a million, right? So 8, 9, 100 million bacterial cells in it. I think I did that right. All right, so let's talk about our next one. Our next type of recombination event is called transduction. And in your book, they talk about 2 different kinds of transduction: generalized transduction and specialized transduction. For this lecture and for the test, or for this course, we're really going to just focus on one type of transduction called generalized transduction. And the reason it's called generalized transduction is that the transduction event itself is general. It's random. The piece of DNA that it picks up and that is picked up and transported to a new bacterial cell is random. There's no special sequence of DNA that can be picked up. So we call it generalized transduction. So if you recall from, I guess, chapter 2 or unit 2, we talked about viruses, and the viruses that can specifically target and attack bacteria are called bacteriophages, or simply a phage for short. And if you recall the life cycle of a phage, it's going to attach to a bacterial cell. It's going to inject its nucleic acid into its genetic material into the bacteria. And then once that happens, the bacteria's genome actually gets chopped up. So this bacteria truly becomes kind of a zombie. It no longer has a functioning set of instructions, and all of its DNA has been digested and is now in tiny little pieces. So the bacteriophage proceeds to make its own proteins, its own capsid, and starts that reproductive cycle. If you recall, this is called a lytic cycle for bacteriophages, where they successfully invade and take over a bacterial cell, and at the end, that bacterial cell is going to burst open and release its content. But something very important happens in the assembly period, and that is that when these nucleocapsids are forming, they're going to form around that viral genetic material, whether it's DNA or RNA, depends on the type of phage it is. But sometimes on accident, that nucleocapsid can form around a small piece of the bacteria's own DNA. And when that happens, this phage here is no longer really a viral phage. What we're going to call it now is a transducing particle, because it doesn't have the phage instructions in it. It has a small piece of DNA from that bacterial cell. And again, chances are that little piece of DNA means nothing and does nothing, but we're going to have that 1 in a billion chance that this phage particle now is carrying something of value. So let's watch a quick video. I feel like these videos hopefully are helpful to you on how this actually works. going to work. Come on.
In generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called a bacteriophage, or phage. The phage attaches to the bacterial cell and injects its nucleic acid into the host cell. A phage enzyme is produced that breaks down the host DNA into smaller fragments. Phage DNA is replicated and phage coat proteins are produced. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material.
So we can see that in this way, we have now we haven't done it, right? I guess the virus itself, the phage itself, successfully introduced new DNA into a bacterial cell. And so this is technically recombination. So when that transducing particle goes on to the next bacteria, it's going to, instead of injecting viral DNA or viral nucleic acids into that bacteria, it's going to inject bacterial DNA. And if that bacterial DNA can recombine with the chromosome of that new bacteria, we now have successful recombination occurring. So in generalized transduction, all nucleic acids, or all DNA, I should say, have an equal opportunity or probability of transfer. So this event is truly random, in that really most of the time what happens here is that these new bacteria, or these newly infected bacteria, are going to receive a bunch of garbage. Now again, there is a different kind of transduction called specialized transduction, and as you can imagine, that form of transduction will carry a specific type of DNA over to that new bacteria. And it's actually kind of a cool thing, a cool process, but it's a bit complicated to try to explain online. So I'm going to just leave it out of this unit. So our second type of recombination, transduction. Here, let's ask a question. I think I have a little multiple choice question. Oh, here's a nice question. So are these new bacteria considered recombinants? What do you think? Well, of course they are. Yes, absolutely they are, because they contain new DNA. They contain DNA that they did not have before this recombination event took place. Remember, recombination, the definition of recombination is the introduction of new DNA into a chromosome. So here we have an original, this teal color was the chromosome of this bacteria, and that purple piece of DNA came from here, right? From this bacteria that was infected by this phage 20 minutes ago, or however long ago it was. And now this new transducing particle is going to go to the next bacteria and introduce new DNA to it. So of course, this is considered a recombinant. Chances are this recombination event didn't really lead to anything productive. But again, you have that 1 in 100 million, billion, zillion chance that transduction could lead to some incredible survival advantage for this bacteria. All right, so pause the video, ask yourself this question, and see if you agree with my answer. What is the answer? Specialized transactions, sterilized. Okay, let's see here. So let's start from the bottom up, and let's not talk just about what the right answer. Hopefully you could see straight away that the correct answer is A, but let's start at the bottom with answer number D, or answer letter D, and talk about why these aren't correct, just as a nice little review here. So transformation involves the insertion of DNA into the chromosome, while transduction does not. Well, they both require this, right? Remember, in order to be considered recombination, transformation and transduction are both types of recombination. In order for that to occur, that DNA has to be a part of the new organism. So whether or not that happens through transformation or transduction doesn't necessarily matter, but it has to have new DNA introduced to the chromosome. let's see, transformation is specialized and transduction is general. Transformation is completely random. Remember that it's literally just kind of DNA floating around in the media that may or may not get taken up by a competent cell. So transformation is completely random. Transduction can be random, it can be general, or it can be specialized. The only mechanism that we discussed was the generalized transduction. Letter B, transformation is the transfer of DNA by phages, and transduction is transfer naked. Of course, these are switched. Transformation is transfer of naked DNA, whereas transduction is transfer of DNA by phages. All right, so that pretty much wraps it up. Very nice short little lecture for this middle part of lecture number 9. And to wrap up our we have a third lecture, and I think I'm gonna do CRISPR as our fourth lecture. Remember, we're going to talk about conjugation, and there's 2 types of conjugation that we're going to talk about, and we're going to talk about some fun experiments, so gear yourself up for that third lecture.